Good afternoon, everyone, and welcome to the CGC July webinar. Today, we'll have a presentation by Dr. Nadia Lanman. She's a research assistant professor at the Department of Comparative Pathobiology at Purdue University. She's also directing the Computational Genomics Shared Resources and managing the Purdue side of Collaborative Core for Cancer Bioinformatics. Dr. Lanman's work at the Cancer Center focuses on managing bioinformatics core, training, and data analysis, and her lab's research is focused on utilizing large genomics data sets to expand knowledge of molecular bases of cancer as well as immune and inflammatory disease. Dr. Lanman is particularly interested in data integration and in developing methods for data sets that leverage temporal or spatial resolution. So today she will give us a presentation entitled A Comparative Analysis of the Molecular Characteristics of Canine and Human Gliomas. All right, you can take it away. If you guys have questions, please do post them in the chat, and then we will have time at the end to cover your questions to Dr. Lanman. Thank you.
Fabulous. Thank you so much. So thank you for having me today and for the wonderful introduction. And so I'm excited to talk about this project. In addition to this being a really fun comparative analysis of a number of large data sets, which is really made possible by the interoperability of the 7 Bridges ecosystem. Really, this has been a really fun project too, in terms of it's truly team science. So I am not an immunologist, I am not a glioma expert, and yet I have an exceptional team that we've all been working together on this project. And so, we've been sort of moving it forward in that way. So I have a number of folks that I'll be thanking at the end as well, who are really to thank for the work we've done on this. So today I'm going to be talking about a comparative analysis we've been doing of molecular characteristics of canine and human gliomas. And gliomas are a common type of tumor originating in the brain. So about 33% of brain tumors are gliomas. And they're the most common type of primary malignant brain tumor. And they originate in glial cells, which surround and support neurons in the brain. And so the 2 types of glioma that we'll be primarily focusing on today are astrocytomas, which develop from astrocytes, which are glial cells which support the metabolic processes used by neurons. They transfer mitochondria to neurons, et cetera, as well as then oligodendrogliomas, which originate from oligodendrocytes. And so these are glial cells that really sort of, they myelinate, they wrap themselves around the neurons and support the neurons. And there are other types of gliomas that I won't go into today. They weren't part of the data sets that we've been using. And so we're focusing primarily on astrocytomas and oligodendrogliomas. And as I mentioned, we've been doing a comparative analysis. So we've been comparing canine gliomas and human gliomas. And so after I'm done with this introduction of gliomas in humans, I'll discuss a little bit about the canine model system. And then I'll go into why it's a good system for also studying gliomas and a little bit about canine gliomas. And then we'll get into the meat of the project, where we've been using some public data sets that are really stellar resources that have been shared with the community to try to gain insights on the molecular mechanisms at work in gliomas. And so interestingly, astrocytomas are actually more common in humans. So that's the type of glioma that's commonly seen in humans, and oligodendrogliomas are actually more common in canines or in dogs. So gliomas grow within the substance of the brain, and it often mixed with normal brain tissue, which really can lead to a number of challenges, as you can imagine, in treating gliomas and in excising the tumors from the brain. This is a photo in the bottom right of glioblastoma tumors, which are, it's a type of astrocytoma. And it's many of you have probably heard about glioblastoma. It's a really devastating disease with fairly dismal prognoses. So there are 4 different grades of brain tumors, and the treatment and the need for, so once an operation is performed to remove the mass from the brain, the need for treatment after this procedure depends really on the grade of the tumor. And so it's also important to note that, I mean, in the context of the brain, even non-malignant or non-cancerous tumors can be devastating and deadly because simply of their location. And so, you know, these procedures are really delicate, and then often, you know, unfortunately, the treatments don't often work very well for the malignant brain tumors. So grades 1 or 2 are low grade, and then grades 3 or 4 are considered high grade. So as I mentioned earlier, glioblastoma is an astrocytoma, so it's actually a grade 4 astrocytoma. So it's high grade. And in canines as well as humans, usually gliomas are diagnosed, unfortunately, in the once they've reached at the high grade stage. So, and the grade also is, so the grade is really based on the growth potential of the tumor as well as the aggressiveness. And so, as I mentioned earlier, you know, gliomas are separated into different types based on the type of cells that they develop from. So in addition to the astrocytomas and oligodendrogliomas, there's also another category that's often you can read about in the literature and is actually present in the data that we've looked at, called oligoastrocytomas, or otherwise known as mixed or undefined gliomas. And so, and unfortunately, it's important to note, though, that high-grade tumors have a truly dismal prognosis. Glioblastoma itself, so again, grade 4 astrocytoma, it's nearly universally fatal. And so here's, I have a table of some of the American Cancer Society's survival statistics. And you can see here that the statistics for these diseases are not good. And they do vary by age somewhat. So you have not too surprisingly, a better prognosis many times in younger patients. However, it's still, you know, for glioblastoma, even in the 20 to 44 age range, only 22% of patients, you know, survive 5 years after being diagnosed or enrolled in the study. And for the 45 to 54 age group, that goes down to 9%, and then 6% in the 55 to 64 range. And so of note, anaplastic astrocytoma is actually another way of saying grade 3 astrocytoma. So, and then low grade would be, you know, grades 1 or 2 again, but in our data sets, we have all either grade 2, grade 3, or grade 4 in the human data sets. So, you know, this is a comparative analysis. So we are actually using a data set that captures the molecular profiles of gliomas in pet dogs. And so at Purdue University, for example, we have an amazing vet school, and we often, you know, see a lot of pet dogs that have gliomas as well as other types of cancer. And lately, the National Cancer Institute, as well as a number of labs across the country and the world, have been really seeing a lot of benefits to using the canine model system. So these are, you know, pet dogs that are brought in to the clinic by, you know, by their families, and they've developed cancer spontaneously. And there is still, of course, a place for mouse model systems, and there are a number of pros and cons of each of these systems, so mice and canines. So some of the pros of mice or murine model systems, of course, it's incredibly easy to control the variables, and to control the environment. There's also the availability of genetically engineered mice and humanized mice. And some of the cons, though, is that the environment and housing is not applicable to humans. So, these are animals that are kept in very controlled environments, in cages, and they don't share the same environment as their human counterparts. So there's also very limited options for spontaneous tumors. And as a result, you often have, for example, in patient-derived xenograft mice, PDX mice, they have to be immune compromised. And so there's no ability then to see how the tumor microenvironment, the immune cells interact within the tumor microenvironment with the tumor. Additionally, genetically engineered mice and humanized mice are very expensive. And then they are also too small for most surgical procedures, and it's hard to get blood out of them, et cetera. So some of the pros of using the canine model, they develop spontaneous tumors. So they actually develop cancer at higher rates than humans. They have a shared environment with humans. They have a compressed lifespan as well. And so, and then they're outbred and they have a heterogeneous genome compared to humans. So this actually is exceptional for mapping of traits. They're importantly immune competent. And so they have, these pet dogs that are brought in with cancer, they have working immune systems. And so we can actually study the interaction of the immune system with the tumor. They also have heterogeneous tumor biology and immune responses. Some of the cons are that it is difficult to control the environment and to standardize treatment. There's a lot of variability due to breeds, and we can try to control for that. But in many studies, there's just too small a sample size to adequately control for breed variability. There's also limited standardized reagents available for canines. And then the last con is also the last pro on this chart, is that they have heterogeneous tumor biology and immune responses. So canines, so these dog models do get gliomas at a fairly high rate. So over 50% of all gliomas occur in brachycephalic breeds. And so these are like the short-nosed breeds, such as shown on the bottom here, boxers, bulldogs, Boston Terriers. And there's a 2018 publication that we now use to type and grade canine gliomas. So the incidence of brain tumors in adult dogs is 2.8 to 4.5%, though individual studies vary. And then 36 to 70% of primary brain tumors in dogs are gliomas. So as I mentioned earlier as well, high grade tumors are more common than low grade at the time of diagnosis. And I also mentioned in human, astrocytomas are more common, but oligodendrogliomas do occur. And in canine, it's the reverse. So oligodendrogliomas are more common in dogs, but astrocytomas also occur. And importantly, there are still a lot of gaps in knowledge regarding the biology and molecular characteristics of glioma in dogs. And so, so what we wanted to do in this project was to look at how similar gene expression and mutation profiles are between canine and human gliomas. we wanted to also identify molecular differences between astrocytomas and oligodendrogliomas. So these are usually diagnosed based on histology and also, for example, IDH mutation status, or IDH status. And but we wanted to see if there were discrete gene expression or mutation differences between the two as well. So we were also curious what genes would differ in expression between low and high grade gliomas, and more so even how the tumor microenvironment differs by grade, if at all. We also wanted to see if there were discrete immune cell composition changes between low and high grade gliomas, as well as between astrocytomas and oligodendrogliomas. And so some of the data sets we used in the study are shown here. And so we, have really enjoyed using the Seven Bridges ecosystem because it's been very easy to pull together these diverse data sets. So the first data set is focused in canines. And so it's from the Integrated Canine Data Commons, or ICDC, which is, it's a data commons that contains a number of canine samples that have some sort of cancer diagnosis. And in this case, there were 83 canine gliomas in this data set. And there were 31 astrocytomas, 46 oligodendrogliomas, and then 6 undefined gliomas. So not all of these, this data set had methylation data, mutation data, whole exome, whole genome sequencing, as well as RNA sequencing data. And this first reference here describes the original publication. It's really stellar work. And the authors focus there a lot as well on the methylation profiles. We did also, this data set contains 3 normal adjacent to tumor samples. And so in addition to those, we actually supplemented this with 12 true normal samples from healthy or dogs without gliomas. And so the reason for that is to let us really probe the tumor microenvironment and be more confident that some of the differences we're seeing are actually normal versus glioma differences, and that we're not missing anything because of the possible proximity of the biopsy site in the case of the normal adjacent to tumors to the actual tumor that was removed. And so we used 7 samples that were from Dr. Debbie Knapp's lab at Purdue University. They're true normal samples. So she and Dr. Deepika Dewan provided those and got them sequenced. So we have RNA sequencing on 7 true normals from Purdue, from the Knapp lab. And we also supplemented this with 5 samples, normal samples from BarkBase, which is sort of like a GTEx type data set where you have a number of normal canine samples available. And this that reference is listed here, number 2. So, and then we also compared this data to human data sets. So specifically the Cancer Genome Atlas data set, which contains 1,115 adult gliomas. So 190 of these are oligodendrogliomas, 793 are astrocytomas, and 131 are mixed gliomas. And we also supplemented this data set with 464 true normal samples that we downloaded from GTEx, which has various tissue types, RNA-seq data in various tissues from healthy or basically patients that don't have cancer in humans. So, and then we also wanted to look at, compare with some pediatric data. So this reference the Amin et al. Reference published in Cancer Cell. That was the original publication in the canine glioma data set, showed that, at least in terms of methylation profiles, that the canine glioma seemed to have more in common with pediatric glioma compared to adult. And so we supplemented this with a data set from the Kids First database. So, and it contains 530 pediatric gliomas. And again, far more of these are astrocytomas, 18,000, and then there's 1,260 oligodendrogliomas. I believe that this is a typo. This should be, I believe, 1,800 astrocytomas. So one of the first things we looked at is that, so in the TCGA data, and this is taken from the original TCGA publication in gliomas that's cited on the previous slide. So there, that group found that there are 3 key oncogenic signaling pathways that are often mutated in glioblastoma. So, and these are the PI3K, RTK, RAS pathways shown here at the top, the P53 signaling pathway here at the bottom left of the figure, and then the RB pathway, which is also dysregulated, primarily in terms of copy number variations. And so, so each of these pathways plays a critical role in oncogenesis. And so, and for example, in the case of the RTK RAS PI3K pathway, 88% of these samples in the glioblastoma TCGA data set show a mutation or alteration, like copy number variant in this pathway. 87% have alterations in the P53 pathway, and 78% had alterations in the RB signaling pathway. And so the first thing we wanted to see is sort of out of these canonical pathways that are known to be important in glioblastoma, certainly in humans, whether these were also altered in canines. And so this figure, it shows, it's called an oncoplot, and it shows here, you have on the top tumor mutation burden in all the various samples. So we have 79 canine samples that had mutation data that we looked at. And so, and then, so here in this kind of main part of the figure, you have, we put the genes from all these 3 different canonical pathways, and then we show whether they are altered in some way. So in terms of, for example, in-frame deletion, missense mutation, et cetera. So homozygous or high amplification, homozygous deletion, and so on. And so then on the bottom of this figure is just some annotation bars that show, for example, grade, high versus low, morphology or astrocytoma, oligodendroglioma, or undefined, and then also breed here. And so overall, what you can see is that there are a lot of alterations in these canine samples in these 3 pathways. And so overall, almost 76% of the 79 samples, so 60 samples had alterations in these 3 pathways, mirroring what we see in humans. And so we took a little bit of a closer look at this and actually looked at, out of the kind of canonical oncogenic pathways that are highly mutated, which ones are the top mutated in canines. And we see a number of known oncogenic signaling pathways, Wnt, TP53. We do see these are some of those, the canonical pathways that are known to be important in gliomas or altered in glioblastoma. And so here on the left side of the figure shows the fraction of the pathway, the signaling pathway that's affected or altered, mutated in high-grade canine gliomas. And then here in the middle in blue, this is in low-grade canine gliomas. And then on the right side, you can see the fraction of the samples affected. So these are the fraction of the samples that, for example, have an alteration in the Wnt pathway. So 22 out of 79 samples have a mutation in one of the genes in the Wnt pathway. And overall, we found that 8% of the samples had a TP53 pathway mutation, 24% had a PI3K pathway mutation, and 48% of the samples had an RTK-RAS pathway mutation. And overall, we can also see, particularly on the right-hand side of this figure, that high-grade gliomas harbor more mutations in known oncogenic pathways. And so we also looked at copy number alterations. And so here we used CNV kit package to identify copy number variations. Of note, pretty much every single software that we've used in this presentation, with the exception, I believe, of CyberSort, has been packaged and put as an application on the Seven Bridges ecosystem. So anyone who wants to replicate any of these analyses or use some of these packages on their own data, you're able to. So the previous slide, we used math tools, and so that's up. CNV kit is also up on Seven Bridges now. And so CNV kit uses what's shown on the y-axis here, the coverage log ratio, to call copy number variations. And then we have the genes from these three pathways are shown, the TP53, PI3K, or TK-RAS, and RB pathways are shown here on the x-axis. And these are violin plots. So these are basically density plots of the distribution of copy numbers of each of these genes across all the samples. And so we have colored the density plots or the violin plots for certain genes in either purple or red. So red shows those genes that have a copy number variation seen in over 10% of the samples. And purple shows those that have copy number variations in over 5% of the samples. And then black shows those that are in, under 5% of the samples. And so, and of note, in adult glioblastoma, the most frequent alterations seen were in the CDKN2A gene, CDK4, and RB1. And so, so in this case here, we can see that CDKN2B definitely has a deletion. And then let's see, for example, P10 also has a deletion in a number of the samples, in more than 5, but less than 10% of the samples. And so overall, we are seeing that, the genes in these pathways, much like we see in the human cohorts, are altered, and that we're seeing a lot of mutations and copy number alterations in these pathways. And so overall, what we've seen is that over 65% of the canine samples have a mutation or copy number alteration in one of the genes in this RTK-RAS PI3K signaling pathway. And less so in TP53, so 16% of samples have an alteration in the P53 pathway, and 47% have an alteration in the RB signaling pathway. And so here you have, we have the genes that are in these sort of canonical pathways that we've modeled off the TCGA analysis. And in the left box here, these are the percent of percent of samples that have a mutation in, for example, EGFR. So 12% have a mutation in EGFR. And then the right-hand box shows the percent of samples that have a copy number alteration. So for EGFR, we didn't find any samples that had a copy number alteration. So that's why it's gray and NA. But for example, FGFR had 5% of samples had an amplification in FGFR, and 7% had a mutation. So overall, you know, despite the relatively low sample size that we have here, 79 samples, we are seeing major alterations in these pathways. So we also decided to look at whether we could see immune composition differences between low and high grade in particular, but as well as between normal and glioma. And so we ran the tool CyberSort, which uses basically will estimate the proportion of various immune cells in your data based on RNA sequencing. And so here, what we found, I mean, we do see some non-significant differences in B cells. There appear to be overall more B cells present in normal versus gliomas. However, the differences are not significant. So we do see some differences in macrophages for the resting or M0 macrophages. None of the differences are significant. Same with the M1s, which are could be thought of as like the anti-tumor macrophages. And then for the M2 macrophages, which could be thought of as pro-tumorigenic, you see that there aren't significant differences between low and high grade gliomas, but there is a significant difference between both high grade and normal and low grade and normal. And so we're seeing really, elevated numbers of M2 macrophages in canine gliomas as compared to normals. We also see significantly elevated numbers of monocytes, or proportion of monocytes in normal compared to both high and low grade gliomas. No difference really between high and low grade gliomas, though in terms of monocytes. And this really makes sense because we would expect to see, in healthy brain tissue that there would be, a number of monocytes that haven't differentiated into macrophages yet, and in gliomas we're seeing very low numbers of those. We are also seeing high proportions of neutrophils in both low and high grade gliomas, and this mirrors what is seen in human glioma patients, that you tend to have much higher numbers of neutrophils, and these have actually been shown to play a pro-tumorigenic role in gliomas and sort of enhance the cancer or the glioma stem cell pool. Across the board, we did, of course, we were interested in seeing whether we saw, NK cells. NK cells, or natural killer cells, they're known to not be common in the brain. But, we do see possibly a lower but not significant difference in the numbers of NK resting cells, but we didn't really see any significant differences there. And then we do see some differences in T cells. So we see significantly more CD4 positive memory resting T cells, and then in gliomas compared to normal. And then we see higher proportions of T follicular helper cells in normal compared to glioma. And so one thing to note is it's known in, for example, in human gliomas, as well as it's been thought in canine gliomas, that while you do have sometimes elevated numbers of CD4 positive and CD8 positive T cells, you also have a number of other cellular infiltrates, such as Tregs, that are sort of switching those T cells off. So you have sort of this interplay of immune cells and then also immune suppressive cells that are, basically immune activating and immune suppressing cells. And so what we were hoping to see here is that, we might see elevated numbers of Tregs or proportions of Tregs in these data, but we were actually not able across the board in normal, low grade or high grade. We weren't really able to pick up any signals of T regulatory cells in this data. So we did also run an SSGSEA or gene set enrichment analysis with specific signatures that we were interested in seeing, specifically like immune enhancing signature, immune suppressive signature, as well as the classic MAPK pathway, the stress junk pathway, and then PI3K, AKT, mTOR pathways. And so we were interested in seeing basically whether any of these gene expression signatures were enriched in our samples. And what we can see, so here you have each column is a different sample, and then each row is the gene expression signature in that sample, showing like a normalized enrichment score. Basically, red or orange would be highly expressed or highly activated score, and then blue would be a suppressed or like negatively enriched score. And so, and you can see here that we've annotated tumor grades. So these are all the normal samples here, and the yellow bar at the top depicts these are low grade samples, and then on the right we have high grade samples here. And you can see that MAPK and the PI3K pathway is highly enriched, and it's active in all the samples, including normal. So you have kind of orange colors across, in each of the samples. So these pathways are active in normal samples, but they are much more enhanced in gliomas. Additionally, the junk pathway is active in gliomas, but it's not active in normal samples. So it's got basically an enrichment score of around 0. And then another thing we can take away is if we look especially at the immune signatures, which we're particularly interested in, you can see that there is actually significant heterogeneity seen across samples. So if you look across the rows, you see a lot of heterogeneity between these different samples. And this really exemplifies the strength of using the canine model, is because the heterogeneity seen in these tumors and in the tumor microenvironments really mirrors that seen in humans. And then we can see too, if you look sample wise, so now look kind of in each of the columns here. So you can see that the immune suppressive signature, which is the second signature down, is in general higher expressed than the immune enhancing signatures. So for example, if you look in the first high grade sample here, you can see that the immune enhancing signature is downregulated, the immune suppressive signature is upregulated. And so pretty much across the board, only a few glioma samples have a stronger immune enhancing signature than suppressive signature. And so what we're really seeing here is what's been suggested in the literature, that we do have immune cell infiltrates in gliomas, and we have CD4 positive T cells, CD8 positive T cells, but we're seeing a lot of immune suppression occurring. And that could be part of the reason why some of the immunotherapies are not successful in treating gliomas. So we then wanted to take a closer look at some of the genes in these signatures, and we broke them down into stimulators, inhibitors, as well as ligand receptors and those involved in cell adhesion. Overall, you can still see, again, a lot of heterogeneity between the samples, like massive heterogeneity between the samples. But overall, if you look at the patterns, we see largely that there's a lot of blue, so that there's downregulation of many of these immune modulators in normal tissues, especially as compared to both low-grade and high-grade gliomas, that you have a lot more upregulation of some of these immune modulators in gliomas compared to in normal tissue. And so, for example, a lot of these are known to be immune suppressive. So PD-CD1 is also probably known as PD-1, for example. And so many of these, genes, for example, are much higher expressed across the majority of the glioma samples than the normal samples. And so what we're seeing is, yes, we're seeing some immune activating genes that are on in gliomas, such as CD28 and CD80, but we're also seeing a lot of immune suppression. So we then, we wanted to also focus on what the differences were between astrocytomas and oligodendrogliomas. In the previous figures, we weren't seeing a lot of differences between astrocytomas and oligodendrogliomas in terms of the immune modulators, possibly slightly more downregulation in the oligodendrogliomas, but it's hard to be sure. And so we did a differential expression analysis between astrocytomas and oligodendrogliomas. And so here we've highlighted the top 20 genes, the top 10 downregulated and the top 10 upregulated. And here again, you can see CXCL8. This is a known immune suppressive gene, and it is expressed higher in oligodendrogliomas. So this is on the Y or on the X-axis, you have the log fold change, and we took oligodendroglioma over astrocytoma. And then on the Y-axis, you have the minus log 10, the adjusted false discovery rate. And so here, the red genes are those that are statistically significant, and then the gray genes, the gray points represent genes that are not statistically significant at a 5% false discovery rate. And so, we see also, so the genes here on the left side are higher expressed in astrocytoma compared to an oligodendroglioma, and then those on the right are expressed higher in oligodendrogliomas than in astrocytomas. And so here you can see that, for example, CXCL8, which is an immune suppressor, is expressed higher in oligodendrogliomas than in astrocytomas. PAX3, which you can barely see here because it's in the mix with a bunch of other genes, but this is a gene that is known to be enhanced in expression in glioblastoma. So this is this also mirrors, the human, what we see in human astrocytomas. And these are, again, the genes that are higher expressed in astrocytomas. We see a number of dysregulated pathways that when we do a pathway analysis on these differentially expressed genes. Here on the X-axis, you see the minus log Benjamini-Hodgberg false discovery rate. So it's basically the adjusted for multiple testing. And then on the Y-axis, we have a number of pathways that are statistically significant. As per usual in a pathway analysis, some of these the pathway names themselves don't make sense. Some of the genes within these pathways do, but those that were really stuck out to us were, for example, the wound healing pathway. There's the tumor microenvironment pathway, which had a lot of actually immune suppressive genes that are upregulated in oligodendroglioma. And here we've colored actually these bars by the pathway Z-score, which is basically whether the pathway is upregulated in oligodendroglioma relative to astrocytomas or downregulated. There's, for the regulation of EMT pathway seen here, we couldn't tell, like the Z-score was not, we couldn't tell whether it was upregulated or downregulated because some of the genes expression values were conflicting. So, so we do see the glioblastoma signaling pathway, which includes a lot of like the genes that are in the PI3K pathway as well as MYC. We do see IL-8 signaling is also enriched in oligodendroglioma, which is known to promote tumorigenesis in gliomas in humans. We see that IL-15 production is actually downregulated. So it's IL-15 production is, the genes involved in IL-15 production are expressed higher in the astrocytomas than in the oligodendrogliomas, for example. And so again, we're seeing a lot of immune-related pathways here, as well as, glioma signaling, glioblastoma signaling, which again involves some of those 3 canonical pathways that I mentioned at the early part of the presentation. And then, for example, regulation of EMT pathway, and wound healing is known also to be important in sort of maintaining cancer stem cell population. And so we do see a lot of interesting pathways that are up that are deregulated between oligodendrogliomas and astrocytomas. And so, this is, even though usually the distinction between oligodendrogliomas and astrocytomas is made histologically, largely, there's the possibility, we thought too, that we could also use gene expression profiles to identify some of these samples as oligo or astro, and so on. So we were interested to see whether there are immune or stromal differences between astrocytomas and oligodendrogliomas. So we used a program called Estimate, which basically it's works in kind of a similar way actually to CyberSort, where it uses a panel of genes and tries to assign an immune score or a stromal score to, or immune score and a stromal score based on gene expression profiles to each of the samples. It's important to note that the immune score actually will, it's just basically, it's not how active the immune system is, it's more the infiltrates. So how many, what the proportion of immune infiltrates is suspected to be. So this could not just be immune activating cells, but also, for example, Tregs, et cetera. And, ultimately, we don't see a significant difference in the immune score between astrocytomas and oligodendrogliomas, but not surprisingly, based on what we've seen on the previous slides, we do see a significant difference between normal and oligodendroglioma as well as normal and astrocytoma. And in terms of the stromal score, we also see a difference between normal and gliomas, as well as actually between astrocytomas and oligodendrogliomas. So we did look at whether there were immune cell proportion differences between astrocytomas and oligodendrogliomas. Overall, like we don't see much difference. We again are seeing the differences, for example, between proportions of M2 macrophages in between normal and gliomas, but not so much between oligo and astro. So here again, you can see this is the difference we saw earlier in monocytes. You can see that there's higher numbers of monocytes between in normal samples compared to either astrocytoma or oligodendroglioma, but no difference between astrocytoma and oligodendrogliomas. And then we do see the elevated neutrophils still, low numbers of NK cells, and non-significant differences. And then again, we see the T cell differences that we saw in the earlier slide here, but nothing is significant between astrocytoma and oligodendroglioma. So based on, we did find a number of differentially expressed genes that were different between astrocytomas and oligodendrogliomas. And so we did actually look to see whether we could build a classifier that could classify astrocytomas versus oligodendrogliomas based on gene expression profiles alone. And so initially, an SVM was fit to subset features that could classify the pathologies, astrocytoma versus oligodendroglioma, and a 109 gene panel was used that optimized the classification rate. And then we looked at how many of these were statistically significantly differentially expressed. We took a pretty loose FDR cutoff of 0.25. And then these 65 genes were used in a Naïve Bayes classifier using leave one out cross-validation. And we actually achieved an 88.6% prediction accuracy that we could somewhat accurately, distinguish between astrocytomas and oligodendrogliomas using gene expression profiles alone from these 65 genes. And we have a pretty good F1 score as well of 0.8675. And then this heat map just shows these 65 genes. And then we used hierarchical clustering to cluster the rows. And basically you can see in these 65 genes that there are differences in gene expression profiles that are visible, are obviously visible between the pathologies. So between astrocytomas, which are shown on the left with the blue annotation bar above, and the oligodendrogliomas shown in pink. And so we wanted to actually look at how stable these clusters were and to sort of validate that classifier. And so we used consensus clustering. So basically it calculates sample co-clustering frequency using an iterative approach of hierarchical clustering based only again on the 65 genes that were included in the Naïve Bayes classifier. And we used, we took a K of 2, so 2 different clusters we were trying to assign samples to, and we used 1,000 bootstraps, and we did 80% subsampling. And so here you can see on the right, this is a heat map that shows consensus score from 0 to 1. So 0 is shown in white, which is the samples are never clustered together. And then 1 would be dark blue, which the samples are always clustered together. And we see, we have 2 different clusters that we've grouped these samples into. And interestingly, so only 10% of the samples were unstable. So they would sort of switch, how they were clustered. And these were the same samples that were misclassified by the Naïve Bayes classifier. And so we ended up actually applying the same Naïve Bayes model to using the 65 genes to the human data from TCGA to classify astrocytomas and oligodendrogliomas. We did only use 45 genes out of the 65 because not all of the canine genes had one-to-one homologs in humans. And we still achieved fairly good prediction accuracy, so 81.8%, and an F1 score of 0.8. So a good classification. And so, to conclude, we did identify that key canonical pathways that are known to be altered in human gliomas are also altered in canine gliomas. We found that the canine tumor environment, microenvironment like that in humans, appears to be immunosuppressive. We use gene expression profiles of astrocytomas and oligodendrogliomas to show that there are a number of alterations in signaling pathways, such as immune-related and tumor microenvironment specific pathways that differ between astrocytomas and oligodendrogliomas. However, we did not see differences in the predicted proportions of specific immune infiltrates. We also used a Naïve Bayes classifier that somewhat accurately classifies canine glioma pathologies based on gene expression profiles alone. And then we use that to predict astrocytoma versus oligodendroglioma profiles in human glioma. And so, this also sort of underscores the importance of using the canine model that it's a valid model for studying human gliomas. And then I would most importantly like to thank the team that I've been working with. So this has been a really fun project so far, and I'd like to thank Dr. Sagar Udikar at Purdue University. So he's the assistant director at the core and a senior bioinformatician. He's just done a massive amount of work and put together a number of these applications on the Seven Bridges ecosystem, so that if any of you want to try these packages, they're there and they're working. Dr. Deepika Dewan has been instrumental in this project and developed the Naïve Bayes modeling framework. And then Dr. Deborah Knapp is a veterinarian and oncologist at Purdue University. And she and Dr. Tim Bentley, who is a glioma expert and an oncologist at Purdue, have been instrumental in helping kind of direct this project. And then I'd like to thank the other folks from Purdue as well. So Harish Kathandaraman has recently joined the team and has done a lot of work on this project and put a number of applications up on this and has been processing a lot of the data, as well as Dr. Min Zhang and Danny Liu from Purdue Statistics Department. And then Matthew Byers from Frederick National Lab, who has been instrumental in driving this project. And also, we wouldn't have even, so he's helped us identify a number of the data sets that we used in this. And then not least our funding sources, and then of course the families who donated their beloved pets tissue for these studies. And with that, thank you so much. And I don't know if we have time for questions, but if we do, I'm happy to take them.
Thank you so much, Nadia. That was amazing. We do have time maybe for one question or two. If there's someone attending the call that would like to ask, maybe they can unmute themselves and I can ask the question instead of writing it in the chat. If not, then I do have a question for you. Yeah. There's some studies that recognize microRNAs as potentially being contributors to gliomas. Have you thought about looking at those in your samples?
Yes, that would be very, very interesting. So I think the canine data set, unfortunately, that we're working with didn't look at microRNA expression, but that is absolutely would be an interesting thing to look at. So.
Yeah, I think you didn't mention in your talk that you were going to use some pediatric samples. How do you anticipate that these results would be similar or dissimilar, I guess, to what you found in canine and adults?
Yeah, so based on that Amin et al., the original publication, and they showed that the methylation profiles were highly similar between the pediatric, in particular the pediatric and the canine glioma samples. And so we're still sort of bringing the pediatric samples into the fold. But what I hypothesize is that based on that the gene expression profiles then as well of the pediatric and canine gliomas may have a lot of similarities. more so even than perhaps the adult glioma to canine. And then we're also really interested in seeing, the mutation profiles and whether we suspect those 3 canonical glioma pathways will be mutated and in those samples as well. So.
Yeah, that sounds great. Can't wait for the next step. Yeah, so I did want to say we're at 3. We will have the recording of the website of the talk on our website in the next couple of days. You can find it at cancergenomicscloud.org/webinars. We also have it, we'll have it on the YouTube channel. We do have a question, consider correlating the pathology of MRI images in the ICDC to these genomic analysis is one of the questions that just came into the chat.
That's a fabulous question. So, so I believe at least when we started working on this project, the MRI images were not yet available publicly. And I need to check on whether those are now, but that would absolutely be of great interest. So yes, 100%. We would be very excited. So they're public. Wonderful. So yeah, we definitely want to do that.
Yeah, so if in any case, if at any point there's some tools that you will need to support that analysis, please let them know. We can wrap them and make them available for you, of course, and everyone in the community that wants to use this.
Fabulous. Thank you so much.
Well, thank you so much, Nadia. This was a really great talk, and thank you so much, everyone, for coming. And like I mentioned, the recording will be available on our website as well as the slide deck. And we will have the next webinar will be August, the end of August, the last Wednesday of August, and we'll be covering proteomics and how to run FragPipe on our. So thank you so much, everyone, for coming, and we'll hopefully see you next month. Bye.
Thanks, everybody. Thanks, Nadia.
Thanks. Bye.
Bye.